So, okay, I'm going to start with some water. Okay, all set. Great, so it's really a pleasure. I'm really delighted and honored to be here today, and I'd like to thank the organizers and the Rank Foundation for inviting me to give the lecture today. So I'd like to start today by asking a question with you, and it's a very simple question, but it really poses a big challenge to our brain. And the question is, how do we make sense of the world around us? Now, in our everyday life, we encounter busy and dynamic environments. And the challenge that this poses to our brain is that the brain really needs to put together a plethora of information that comes from many different sources and try and make sense of it. And so how does the brain do it? It's not an easy task. And really what the brain is trying to do is identify features, identify targets of our attention and action, interpret the current scene, but also predict what's going to happen next. And so what I'd like to propose today, and I'd give you some evidence for this as we go along in the talk, is that actually the brain selects and processes very different types of information depending on the goals of our actions. And so, for example, if you found yourself in this cluttered New York City street, your brain would process very different information if you were trying to identify a friend in the crowd somewhere over there. Or if you were trying to hail a free taxi, or if you're trying to cross the road. Now, Nijmegen and Cambridge are much quieter cities, but the problem and the challenge to the brain remains the same. So your brain would process very different information if you were trying in these scenes to identify a friend among the cyclists, or if you were trying to spot a really fancy and smart bike, a speedy bike, or if you actually were trying to cross the road. So how does the brain do it? This is a problem that has puzzled computer scientists for a long time, and we have seen real exciting advances in neural networks that try to simulate and understand how artificial systems could actually learn to detect objects, to recognize them, discriminate them, and also predict what's going to happen next, how potentially they will move, how they will act, so that we can interact with them successfully. Now, at the same time, in neuroscience, we have been developing many different methods that allow us, with state-of-the-art tools, to study how the brain detects, recognizes, and predicts at different scales, starting from the single neuron all the way to local circuits and large-scale networks. And today, I'd like to tell you how we do this in my lab, how we try to understand how the brain moves from our senses, from vision to decision, and how the brain really takes into account different previous experience to interpret scenes and predict the future from the past. And in my lab, since the time that Richard joined us, we tried to put together behavioral paradigms with multimodal brain imaging and with advanced computational methods. And so the main question that we really ask is how the brain learns. What are the mechanisms that the brain has and develops to be able to support our ability to learn in our everyday environments and show adaptive behavior. So in particular, how we do this is usually we build novel displays that we present our participants with. They are unfamiliar with these displays, and we ask them to make some judgments about the stimuli that we design in a context that is rather uncertain, noisy, cluttered, like simulating like what's happening in real-world scenes. And then what we do is we collect both behavioral and different types of brain imaging data, and we model this data to try and understand the link between brain mechanisms and behavior. And what I'd like to propose today, based on the work we've done so far, is that the brain really learns decision templates, and these decision templates are adaptive. They comprise of information that is behaviorally relevant. They comprise of features that are key for our decisions. And these templates reflect the structure of the world that is relevant for our specific actions at a given time. And so it's these really behaviorally relevant information that the brain then can use to interpret successfully and fast the environment, but also predict what's going to happen next. So just to give you an example about these adaptive templates that contain information about the structure of the environment, in our previous work, we have looked into how participants classify novel objects that might look like these, patterns that are embedded in noise that are unfamiliar with. And we asked them to assign them to different categories. And of course, this is totally meaningless when they first engage with it. But models can tell us which are the key features that separate the classes of patterns that we present the participants with. And what we see is that only after training, really, participants can extract the key features that discriminate between two classes of patterns. These are the features that you see here, maximally differing between the red, let's say, category and the blue category. Now, the really interesting result from this work is that we can identify similar templates of information in the visual cortex. And what you see here is that these key features that drive our decisions appear only after training rather than before. So this was the first evidence that we had in information space, both behavioral information space and brain space, that the brain represents not simply every feature that we encounter in a scene, but actually the behaviorally relevant information that supports representations of the structure of the environment that then guide our decisions. And so then the next challenge for us is really to understand what are the mechanisms that the brain uses to build these adaptive templates. But just taking a step back, before I show you our findings so far on brain mechanisms, I'd like to ask the question of, can we actually discover any mechanisms of the brain when we use imaging, non-invasive imaging in humans? Now, I've started brain imaging work back in the late 90s, and I never went back. It was really exciting to be there something like 2 or 3 o'clock in the morning at MGH, because the scanners were clinical, not for research, and for the first time see real activity of the brain analyzed online as we were scanning the participants. So I never went back, but the criticism of that has always been that with brain imaging, we can discover a lot about where in the brain, but not how. So what I'd like to show you today is really examples of how with multimodal brain imaging and working at information space rather than at activity space, we can actually extract a lot of interesting mechanisms of the brain that support behavior. And just to show you the methods that I will be talking through the presentation that we used in different studies, we use methods that allow us to investigate the structural plasticity of the brain, and this is quantitative structural MRI. We use methods that allow us to investigate the functional mechanisms of the brain that relate to information, not only at cortical surface, but actually at cortical depth across different depths in the brain. We look also at functional connectivity to understand how functional interactions between different circuits of the brain support adaptive behavior. And finally, thanks to advances in MR spectroscopy, we can now look at neurochemistry in the brain and relate that to the behavior. For example, we look at neurotransmitters such as GABA and glutamate that are known to be involved in learning and brain plasticity. So with all our tools under belt, we can now ask the question of how does the brain really make sense of the world around us? And that's what I'd like to guide you through in the next 40 minutes, maybe, and ask the question of what are the brain mechanisms that support the brain in its capacity to learn, in order to detect objects in clutter scenes, discriminate those objects, and recognize, categorize them to meaningful classes, and finally predict what's going to happen next from past information. Okay, so I hope you're not falling asleep with the lights down. Let's get started. So let's take our first question. How is it that the brain managed to detect information from clutter? How does it know which are the targets of our attention and action? And we know a lot from behavioral work, particularly in the area of perceptual learning, of actually that practice makes better. Definitely when we practice in visual tasks, visual discrimination tasks, and try to identify displays in noise, we see behavioral improvements. So our job here really was, starting from this seminal work, to be able to understand what are the mechanisms that support this ability to improve in these fine judgments with behavior. And the work I'll be showing you today is work that Zoe Kourtzi did in my lab in the last few years, and to his real credit, this is work that actually was completed during the pandemic. So research must go on. So we devised a paradigm using glass patterns. I'm sure you all know about these patterns. They're really interesting. They comprise of pairs of dots, dipoles, and depending on how these dipoles are aligned, you can see that they can form different types of patterns, radial patterns or concentric circles. And then we introduce noise to these patterns, and you can see that here we have only 25% signal really, so this is a really difficult task. We ask our participants to tell us whether they can see a concentric or a radial pattern. Now, what we do is we scan the participants in 3 different occasions here while they're doing this task. The first time around, we collect a baseline measurement without giving them any feedback in the task. Then we wait for 3 days. Then we bring them back and scan them again. And then another 3 days, but during these 3 days, we bring them in the lab and we give them training with feedback on this task. And then we do a final scan where we test them again without feedback. And if you look at the behavioral data, you get this improvement in their performance. It's very clear. When they start, the task is really hard, but through training, they actually improve a lot. So now we have the brain data from these 3 scans. We use the first 2 scans as control, the comparison between the baseline and the pre-training, and we look for differences between the pre-training and the post-training. And so the first scans that we looked at here are structural scans, and we model the data from different types of structural MRI scans to extract information about myelination. So we extract with quantitative MRI myelin content. And we did this across the whole brain. And to actually our surprise, I expected that maybe we will see something in visual cortex. But actually, where we saw the most significant changes were in the thalamus, and primarily in pulvinar. Now, I knew something about the pulvinar, and I had to do my reading again. And we know that the pulvinar actually regulates processing in the visual cortex. So that turned to us as a very interesting development of this project. As you can see here, we have increase in myelination in the pulvinar. So given the role of pulvinar in regulating activity and processing in the visual cortex, we then asked whether learning actually changes the connectivity between pulvinar and visual cortex, and we use functional data, resting state fMRI data. We model this data, and what we see is that training alters the connectivity, the functional connectivity between pulvinar and visual cortex, and these thalamocortical connectivity changes relate to behavioral improvement. Now, the next real question for us was, what happens then in visual processing? And in this case, we looked with MR spectroscopy changes in neurotransmitters like GABA that we know that they are involved in perceptual decisions, but they are also involved in learning and brain plasticity. And what you see here, we had identified this from previous work, is that we know that over time, through training, GABA actually decreases, which is really interesting. Decreases in GABAergic inhibition have been associated with brain plasticity. And it's a very much task-based mechanism here we see that for detecting patterns in noise, and we have seen this in previous studies, we see a decrease in GABA that relates to behavioral improvement. And we think that potentially this relates to a local gain control mechanism in visual cortex in terms of higher reduced inhibition allows higher activity that allows us to detect targets of our attention and action in the environment. And what you see here is that this decrease in GABA also correlates with behavioral improvement. Now, to go one step further and ask whether there is potential a causal link between GABA and behavior, we worked with interventional methods. For example, in this case, we work with TDCS. And what you see here is that actually TDCS decreases GABA over training, primarily during TDCS, but increases behavioral improvement with learning. So we see clearly that GABAergic inhibition plays a role in learning. So far in this study, I've shown you evidence for three types of plasticity. One is structural plasticity, primarily in the thalamus. The other is changes in thalamocortical connectivity between pulvinar and visual cortex. And finally, changes in visual cortex GABAergic activity as a gain control mechanism. So now the question is, how do we put all of this together? What is the circuit that supports actually our ability for learning? And to do this, we turn to modeling. And what you see here is a mediation type of analysis, where what this model tells us is that actually changes in myelination in the pulvinar, in the thalamus, drives the connectivity to visual cortex, and that drives the local processing, the changes in the GABAergic gain control mechanisms. But do these interactions predict behavior? To understand this, we built a structural equation model. And what you see here is that interactions between functional connectivity, thalamocortical myelination, and local GABAergic processing actually predict behavior quite strongly. So we think that what's really going on here is that myelin plasticity in the thalamus interacts with GABAergic processing in the visual cortex through thalamocortical circuits, and that supports our ability for perceptual decisions. So that's briefly to summarize, in the context of learning to detect targets in noisy environments, we seen that the mechanism really that can support this, the adaptive mechanism, is thalamocortical interactions that regulate gain control in visual cortex in order to support improved perceptual decisions. So moving on to the next process, once a brain has detected features that are relevant for a specific task, then recognition comes. We need these features to be able to recognize objects. So how does the brain do it? And this is work that Kezia, Elisa Zamboni, and Menshin Wang have done in the lab. We started before the pandemic and managed to get through the pandemic with this work. And in this case, we ask the question of what is the fine brain architecture that supports our ability for recognition of objects. And there are many different theories that have been put forward in terms of how we really learn object categories. One is that we process low-level features, and in a hierarchical fashion, our brain increases the complexity of the features that are processed. The other is that we have top-down processing from abstract representations, concepts, and categories that influences the visual processing. Now, disentangling these two mechanisms has been the topic of great debate in cognitive and visual neuroscience. And here we have taken a new approach that relies on ultra-high field imaging and imaging across cortical layers. We know from anatomy and physiology that there is an interesting hierarchical structure across cortical depths. and that relates to feed-forward versus feedback processing. So, for example, we know that in primary visual cortex, we have input coming from the thalamus and the middle layers, and that's where the input processing is happening. And then we have long-range feedback from higher visual areas to the deeper cortical layers in primary visual cortex. But interestingly, in superficial layers, we have both long-range feedback, but also local cortical circuits that provide recurrent mechanisms for processing in the superficial layers. And we can disentangle these layers, and I'll show you how, and then ask the question of does processing and does learning, is learning supported from processing in these different layers. We can also ask the question of how these layers are connected with higher areas. So we know that information from superficial layers going into the middle layers suggests feed-forward processing, while information going from deeper to deeper layers suggests feedback. There's also connections between superficial and middle layers and deep layers, but this is both ways, it's both feedback and feed-forward. So we concentrate either in superficial middle, which is feed-forward processing, or deep to deep, which is feedback processing. Now you could say, can you really do this with fMRI? How close can we get? We can get quite close with high-field imaging. This is data collected at the 17 Cambridge, and the resolution here is 0.8 millimeter. So our standard fMRI resolution is 2.5 millimeter, but at high field we can push this resolution to 0.8 millimeter and therefore be able to separate voxels across cortical layers. Now the way we do this is by effectively looking at all the voxels between the PL and the white matter, going from the surface to deeper in the brain, and assigning them weights in terms of how much potentially these fall into different cortical layers. Now we cannot see anything in layer 1 because this is primarily kind of a contribution from large veins, so a lot of vasculature artifacts, but we can then bin the activity we have in these voxels in superficial, middle, and deeper layers. We have to deal with vasculature artifacts. If you look at these signals across cortical layers, you will see the pattern here shown in the blue bars, and clearly the activity is stronger in the superficial layers because we have vasculature effects, but when we clean the signals, we can then start with activity that is similar across the cortical layers. So the work that we did here, actually, we started with a very simple paradigm. These were really our first attempts in the early days. We were going to move much faster into more complex patterns and objects, but the pandemic slowed us down a little bit. We started with a classic perceptual learning paradigm, which is orientation discrimination. It's not anymore detection from noise, it's really fine-tuning of a simple feature, an orientation feature, and of course, the task starts quite hard. So we collect data in the scanner when the participants are doing this task the very first time. Then we bring the participants in the lab and train them, and then we put them back in the scanner and test them again. And I'm sure this data is not surprising. What we see is based on thresholds. We see a decrease in the thresholds over training, and that means the sensitivity in this task is improving. And this effect is very much dependent on the trained orientation. Specific to the trained orientation, we see very little improvement for the untrained orientation. Now, how can we extract information that have to do with orientation signals in the brain? We know that neurons that encode orientation information are actually at a much higher resolution than even 0.8 millimeters. So we can't really look at these signals that are coming from very selective neurons. But to our rescue comes techniques of multivoxel pattern classification that employ linear support vector machines that allow us to learn correspondence, to learn associations between MRI data and labels. We train the classifiers with a lot of data. We leave independent data out, and we test for each new data set that we give to the classifier whether the information in this brain data comes from the trained or an untrained orientation. So here's what our data looks like. This is primary visual cortex that you see here. We segment these into cortical layers. And for each one of these for all of the voxels being in each one of these cortical layers, we take a pattern classifier and we train it to learn and to discriminate between the trained orientation and a control orientation, or between an untrained orientation and a control orientation. Now, a lot of work goes here into the psychophysics of this, into a properly randomized design, separating the trained from the untrained orientation, randomizing across participants. And what you see here is that after training, this is the blue bars, after training, we have an increase in information from primary visual cortex only for the trained orientation, and only in the superficial layers, not in the middle or deeper layers. So this suggests to us that actually what learning is changing is not the encoding, the information at the middle layers, is not the long-range feedback that is coming from deeper layers in higher areas, but it's actually some local processing that is going on in a recurrent manner, potentially a local circuit in visual cortex in superficial layers. Now, this is really interesting because we know that eyes orientation suppression is most prevalent in superficial layers. And therefore, it's likely that this information that we see to be shaped to become more tuned for the trained orientation relates to eyes orientation suppression, which we know that lateral interactions terminate mostly in the superficial layers. Now, we ask the question of whether this effect is specific to the trained orientation. So we take our classifiers, and we also test whether the classifiers can separate an untrained orientation from the control orientation. And you see that before versus after training, there is no significant difference in any of the cortical layers. So the effect is very much specific to the trained orientation. Now, a signature of perceptual learning is also the specificity in location. So we also tested that. You see that the effect in the superficial layers is very much specific to the trained location, but not to representations of either the trained or the untrained orientation in an untrained location. So very much a very specific effect that links to what we know about perceptual learning. So this all is happening in the primary visual cortex, but we didn't want to stop here. We wanted to understand how then the visual cortex provides that information that comes from sensory plasticity to influence decisions that we know are happening in higher up in other circuits. And for that, we looked into the posterior-parietal cortex. And we couldn't really go into frontal cortex because of the way that we put our slices to cover part of the brain. When we scan a 0.8 millimeter resolution, you can't cover the whole brain. So there's a bit of a trade-off. But we placed our slices in a way that covered visual cortex and also posterior parietal cortex. And we know that posterior parietal cortex is involved in decision-making and perceptual decisions. And therefore, we asked the same question: can we extract information from posterior parietal cortex that relates to the trained orientation? And what you see here is that we extract this information, but in this case, in the middle, not in the superficial nor the deeper layers. Now, this to us suggests that potentially what we have here is a circuit where local recurrent processing is happening in visual cortex in superficial layers, and then the signal is sent forward to middle layers in the parietal cortex to support our ability to make perceptual decisions. How can we test this? We went to test this with functional connectivity analysis in this information space. So in this case, what we went back into our classifiers and interrogated the patterns and the signal of the patterns that went into the classifiers and looked at what is their distance from the hyperplane in the classifier architecture that separates the representations of the trained from the untrained orientation. We then took this distance and computed it over time and looked at this kind of information time series between the visual cortex and the parietal cortex. And so in that way, we looked at connectivity between primary visual cortex and IPS. And what you see here is actually that this connectivity was stronger for the train orientation between the superficial layers of V1 and the middle layers of IPS. And there was no real difference in the feedback processing. So from deeper to deeper layers, from deeper IPS to deeper V1 layers, we didn't really see any significant changes in connectivity. So these results really so far suggest that what we have in visual cortex is recurrent processing, and then that information is read out by parietal cortex to make perceptual decisions in a feed-forward fashion. Now the next step, we really wanted to dig down a little bit more and interrogate what happens what is the mechanism that supports this retuning of the orientation, what makes more selective the orientation representations through training. And so we turned to GABA, and we looked at GABAergic plasticity in the context of this task. And this is work that Polly Frango and Vasilis Karlaftis have done in the lab. And when we looked at what happens to GABA during training, in this case, we see increase in the GABA during training in this orientation discrimination task. Now, this is in contrast with what I showed you earlier, which is decrease with training for detecting in clutter. And this pattern we have replicated now a couple of times, and we think that what's potentially going on here is that when the brain needs to shape representations, it needs to become more selective, cortical inhibition increases to support this retuning of representations during training. We've also looked at what happens at connectivity, and we see that this increase in GABAergic inhibition relates to connectivity between visual cortex and IPS. So all of that really suggested to us that GABAergic processing and inhibition play a critical role in shaping this retuning that is happening to support our perceptual decisions related to fine feature discrimination. So we went ahead and repeated effectively this study with high field imaging. And it has been one of the brightest moments of this pandemic for me that we replicated our superficial learning effect in the domain of fMRI. But together with fMRI, we recorded GABA. And what you see here is that in higher GABA during training relates to increased bold activation, but even more specifically, it relates to increased classification accuracy, suggesting that what we have here is increasing inhibition relates to this retuning of the representations. And interestingly, it also relates to connectivity between occipital and parietal cortex. We still have a little bit of work to do with modeling this data, but I think we're very excited that we're getting closer to understanding these cortical excitation inhibition mechanisms and how they contribute to perceptual decisions. So to summarize so far, I hope I've given you some key studies from our work that propose that actually the way the brain puts together information about selective features is by recruiting these recurrent circuits and passing on information that is read out by decision areas. And so to summarize, when we look at going from vision to decisions to perceptual decisions, we see first thalamocortical circuits and interactions that support our ability to detect targets in clutter, and then for fine discrimination of this information that leads to recognition of objects. The information is taken further by occipital-parietal circuits that through recurrent processing consolidate these very selective feature representations that then drive and improve our decisions through training. So the last part of the talk I'd like to dedicate to another kind of strand of work that we've been doing, which moves from our ability to interpret the current scene to actually predicting what's going to happen next. And these things in our brain and our everyday behavior happen seamlessly without effort. We always try to interpret not the past, and almost the present becomes the past, but what we try to do is really move into the future, predict whether we can cross the road or not, predict whether the person next to us are going to be friendly, or whether you're going to ask me some good and friendly questions or not. There's a nice quote here from Niels Bohr. It's difficult to make predictions, especially about the future. Who knew that Britain will have Brexit and a pandemic and drought? Who knew we will have summers? Anyways, this is really nicely documented by these wonderful photographs by Edgerton. The milk drop is one of my favorites. And motivated so from this question, we've looked at previous work to try and understand how short-term history actually can change the representations in the brain. We devised a simple paradigm where we present our participants with gradings of different orientation. Then there was a pause in a sequence of gradings, left, right, and then a test orientation. And we asked the question of actually what happens during this gap. The brain doesn't see any information, but can the brain predict? And interestingly, what we saw was that after training, we have increase in the information in visual cortex that allows to make the prediction of the expected stimulus. We've taken this work quite further to answer the question of how actually our brains learn predictive statistics. How do we interact with the environment, and without feedback, without having a teacher to tell us this is right or not, we learn the statistics of the environment. How do we learn complex structures when we navigate in a new city, or when we try to learn a new language or learn music? And so to try and understand how really our brains pick up now, in this case, temporal structure, and how do they learn the statistics that allow us to make predictions about what's going to happen next, or how we should move and navigate next, or what we will hear next. We built this paradigm where participants are exposed to sequences of symbols. These are unfamiliar symbols. And at the end of the sequence, we ask them to make a choice, which one of these 4 symbols will come up. We don't give them any feedback. This is done in a totally unsupervised way. It's a statistical learning paradigm where the participants are trying to pick out the statistics. And the statistics we determine, they are probabilistic, and they depend on Markov models that increase in complexity. So what we have here is an adaptive training paradigm where participants start learning about just frequency of some of these symbols. Some are more frequent than others. And then their complexity increases, and our participants are asked to learn about pairs and the probability with which these pairs appear, or even triplets and the probability with which these triplets appear. And we don't tell them any of this. We present them the sequences and we let them kind of figure it out. It is a quite hard task. The participants have to actually first figure out the structure. So am I learning pairs? Am I learning triplets? But also then they have to figure out the exact contingencies, which symbols go with with what. They actually do quite well. You can see as the level of complexity increases, we have more participants that actually do not improve. This is with the gray circles, but most of the participants really improve. But really, the interesting thing is what happens to this learning. They learn just the specific symbols and the specific sequences, or can this generalize? Because if actually what people pick out is the structure, the underlying temporal structure, these Markov models that we impose to the sequences, then we want them to be able to generalize. So we ask the question of, can the participants do it when we have new symbols, completely different symbols when we test them? Does the learning generalize when we put in a completely different design? Does it generalize when we change both the symbols and the design? And the answer is really that when the participant learns these structures, it doesn't matter what the symbols are, it doesn't matter what the design is, they very quickly identify a new structure and new symbols because they have learned the structure that is imposed in these sequences, and they can make predictions. Now, believe it or not, when we started this work, what we wanted to look at is brain circuits and brain networks that are involved in this type of learning. That they allow us to they allow us to learn these more kind of complex structures and how we move from more simple to more kind of complex statistics, and how the brain learns these different statistical properties of the environment. But when we looked at the behavioral data, we saw this actually striking pattern of individual variability. And this was something that really we couldn't ignore. In the past, we used to look at individual variability, but most of the times kind of average or remove outliers. Now, actually, with modeling, we can look into this individual variability. And actually, this work has taken us to look more into why some participants, actually, when they try to learn this structure in completely unsupervised manner, learn faster, while some others learn slower. And so we decided to model the data and look at decision strategies. Now, there's a lot of work in animal literature in reinforcement learning that talks about two different strategies for kind of problem solving and decision making. One strategy is matching. This means that when animals and humans have to deal with probabilistic statistics in the environment, what they try to do is match their choices and their response to the statistical regularities in the environment, to the probabilities. So if A is presented 80% and B is presented 20%, then they try to approximately kind of split their choices between 80 and 20%. Another strategy is maximizing. And this strategy is primarily seen when individuals get trial-by-trial feedback. And that means if A is presented 80% of the time, actually, if you choose A 100% of the times, you maximize the outcome, you maximize the reward, because you will definitely then get 80% of the times correct. And it's interesting to see actually how people switch between these strategies depending on the uncertainty and the complexity of the environment. So we model the data to look at these strategies, and here's what we've seen. The colors here actually reflect proper clustering analysis. But just for kind of purpose of explaining, if you look at the red people that learn really fast, you can see that their strategy actually differs from the strategy that the blue folks have who learn a bit slower. And you can barely see here there's a dotted line. This is the matching line. This is when the participants adopt the matching strategy. And what you see is that actually the people that learn this task faster, the people in red, are the people who start moving away from the matching strategy to maximizing. So instead of trying to learn exactly the statistics of the sequences that we presented them, they actually start moving towards maximizing. And interestingly, there's a strong correlation between how fast people learn and the strategy they take. So instead of looking at brain circuits that maybe are involved when we learn simpler versus more complex statistics, we changed path really, and we use this strategy as a regressor in the analysis of both functional and structural imaging data to try and understand what are the networks that support this individual variability in learning and support these decision strategies. And this is work that Ray Wang did in my lab together with Vasilis Karlaftis and Joseph Georgiou. And so we looked here both at structural and functional plasticity and circuits. And what we see is that when people took a matching strategy, the circuits we recover involve the visual cortex, the hippocampus, the caudate, and the thalamus. And this is in reinforcement learning, is the visual loop that involves visual cortex and subcortical regions together with hippocampus. Now, in contrast, when people took the maximization strategy, what we've seen, again through modeling the data, is that this strategy is supported by circuits that involve primarily frontal circuits and putamen, and this is known as the frontal striatal loop in reinforcement learning. Now, there is no reinforcement learning here. The participants do not get trial-by-trial feedback. They are learning spontaneously the statistics while trying to do this task, and we recover the same circuits that we see in reinforcement learning. And really what was surprising for us is that different strategies recruit different circuits. People get to the same point. Everybody learns, but with different rates that relate to different decision strategies. So using the computational modeling to guide the analysis of both the structural and the functional data, we derive different circuits. And just quickly to show you that we recover the same circuits when we do DTI analysis. This is the visual loop that we recover relating to matching, and the frontostriatal loop that we recover related to maximization. And similarly, when we look at resting state functional connectivity, similar, we extract the visual loop for matching, the frontostriatal for maximization. So again, through different signals in the brain, we extract the same circuits that support our decision circuits really and our decision strategies. So to summarize, really, what we have here is cortical striatal loops that support our ability to learn predictive structures without a teacher, without reinforcement learning, and support our ability to make predictions from the past to the future. Now, you could say, why is this important? It's actually really interesting in several cases. In older life, in normal or pathological aging, we see that one of these routes might actually be impaired. We know that hippocampal circuits are impaired in even early dementia. And so potentially here the question is, can the brain reorganize? Can the brain learn a new strategy? Use the alternative route, the alternate route from the route that is impaired, and still be able to learn and predict in complex environments. And with some work that we've done with individuals that have mild cognitive impairment, we actually see that they can succeed in these tasks. They can learn these complex sequences. And interestingly, they use the frontal striatal circuits rather than the hippocampal circuit that we see being more recruited in healthy individuals in later life. So 6 o'clock, I think time to summarize. Thank you for persevering and bearing with me. Back to our big question, how do we make sense of the world around us? I hope today I have shown you how vision is really in decision. It's really integral and key process of decision making. And I'd like to emphasize again that our work really shows that the brain learns not just simple features across scenes, but learns the behaviorally relevant information that allows us to extract structure from the environment that is key for our ability to interpret and predict. And through modeling of both behavior and brain imaging signals, we can now start understanding the circuits across the brain that support our ability for adaptive behavior, starting from thalamocortical interactions for gain control mechanisms that allow us to detect key information critical for our decisions in the environment, and moving on to occipital-parietal circuits of decision-making that support our ability to refine the representations and recognize objects, and finally to cortical striatal loops that support our ability to predict the future. I'd like to close by thanking you and thanking my lab that has made this dream come true. They're a super talented and great and creative team. You can see they're all taller, and I can reassure you, smarter than me. I'd like to thank our funders and also all our collaborators. I could fill up slides and slides with our collaborators. This is not an effort that rely on a single team. It's a very much a cross-disciplinary effort from MR physicists all the way to computer science and engineers, biologists, folks doing animal and human work. I'd like to thank again the Rank Foundation and thank you for being here today.
Well, thanks, Zoe, for this fantastic and inspiring lecture about your work. Okay, so I've here two microphones, so now we can get some answer to the question that you posed in your lecture. So, are we going to get some very nice and friendly questions, or not? So maybe let's start with the nice and friendly questions. Maybe, I don't know. Yes, so here in front.
Don't do the lights. Yeah. Oh. All right,
okay. Oops. It's a little intimidating to ask a question after such a wonderful talk, but here it goes. So you're concentrating on object detection, so very low level, and it's beautiful because you see it in V1. I wonder if you could speculate, would such a mechanism work for much bigger aspects like a scene recognition or something that requires more connection with higher level categories rather than just orientation?
Great, thank you. Yeah, great question. That's why all my reviewers say, oh, this is too low level. No, I know. I'm only kidding. It's a very good question. As you know, kind of going back in the brain and to simpler displays really allow us to have very good control of the stimuli, be able to titrate. And also we have really good animal models to understand where we are. And especially with looking at kind of across cortical layers, we know much better the structure of V1, how we can unfold the brain and look at the layers. So some of these decisions, when we build our paradigms and our tasks, are made based on cutting-edge methodologies that we have, but also existing models. So we take it step by step. As you say, the more interesting questions is really what happens higher up in the brain. And we have looked, and there's a lot of work going on all the way to inferior temporal cortex, looking at object complexity. And it'd be really interesting to be able, for example, to look at cortical layers and inhibition in these higher areas. And we have done some work. What we need is to be able to kind of tie all of this up to be able to understand the mechanisms. Now, I'd say it's my belief, because of previous work, that the brain is actually constructed on the basis of principles. And these principles guide the processing across the whole brain. For example, take retinotopic mapping. Right? We used to believe that this is just a visual cortex, that is a primary property of V1. But we have seen retinotopic mapping in parietal cortex for eye movement, for attention. We have seen retinotopic mapping in frontal cortex for working memory. So I kind of strongly believe that the brain is structured on the same principles, but they express themselves differently in different centers of the brain to be able to support, of course, different task complexity, different behaviors. So slowly, hopefully the new generation, will move into, now that these methods are becoming, more kind of feasible to do, we hope to be able to apply those in more kind of complex tasks and scene recognition. That will be really amazing, because that's where really all of the complexity of the information comes to play. And again, I'd like to emphasize the role of modeling here. Sometimes, we turn to a reductionist approach in our paradigms to be able to control. But with modeling, we can put in more parameters and look at the kind of disentangle the process associated with different parameters. Again, of course, there's quite a lot in trade-off of how we build these models to ensure that they are valid. But we've seen great potential in computational modeling coming from different approaches that I'm hopeful then, when this applied to brain signals, can allow us to extract processes for even higher level constructs in the brain.
Thank you for an interesting talk. I was wondering if you could clarify a little bit. You were talking about a mode of inhibition versus a mode of disinhibition in terms of GABA, depending on whether you're doing fine-grained discrimination, and the other one you labeled as detection. But I would kind of label as detection something where you're sort of operating a threshold, and then of course disinhibition is helpful because that single unit that is most sensitive. But here it looked more to me like a sort of template sorting task where you really have suprathreshold stimuli and you have to decide which category it belongs to, and the stimulus is much more complex than the line stimulus. And could it actually be, rather than inhibition versus disinhibition, some sort of fluency also operating in learning where you're just getting more fluency and therefore less inhibition and the one you labeled as detection?
Yeah, that's really interesting. So maybe I wasn't very clear. The task that I broadly talked about as detection is a discrimination task in noise. So the stimuli are actually not clear at all. There's only 25% signal in the stimuli, so effectively you see random dots. And of course with training you become better. So that's where we see overtraining, the GABA decreasing. We do these tasks in the scanner where people are learning and they're getting better. And we record GABA during this training, and we see this decrease in the GABA. But the stimuli remain. We cannot do really threshold tasks because these are constraints in terms of how long we can keep people in the scanner. But the stimuli are not suprathreshold.
They are in terms of contrast.
On contrast.
Luminance contrast.
Oh, yeah, stimulus contrast, yes. Yeah. So in this case, because these stimuli are the patterns that you saw with the dipoles, the glass patterns, we vary not the contrast, is not a grading, but we vary the stimulus noise. I mean, it depends really on kind of the perceptual salience. That's what that is changing in the displays, and we see that really the GABA decreases in this case. So we haven't done it with contrast detection, although we do have a study looking at this, and it will be interesting to see whether we have a similar mechanism in this case. Another, it's quite interesting how potentially different stimuli might drive different processes and a different kind of hierarchy in the brain.
Hi, thank you for the wonderful talks. Do you think that this pathway that you found for each step can work also with other sensory modality, like instead of V1, the sensory motor cortex, the auditory cortex, but anyway that the circuits are always the same, or you think that's something related to V1?
Okay, great. So are you thinking like, for example, auditory stimuli, and for example, looking at primary auditory cortex versus, I always say, the only thing I know about audition is anything relevant to vision, but that analogy doesn't work. I think, I mean, there is actually a lot of work looking in auditory cortex. I'm not sure actually if people have done looking at kind of cortical depth in auditory cortex, but I want to say that they have. But now I'm really sorry if I'm forgetting relevant work on this. But I think that in other modalities like audition, like, for example, touch, we do have sensitivity in discrimination, for example, discriminating in touch. Right? And we know that there is, in sensory motor cortex, there are similar maps where you can differentiate digits in high-field imaging. So I think that similar principles could apply. And looking into learning, for example, we know that GABA relates to sensory motor learning very much. And we see this decrease in GABA when people are becoming more sensitive in sensory motor tasks.
Okay, thank you a lot.
Okay.
Thank you for the talk that you've given us. I just wanted to ask a question regarding the GABAergic responses that you've seen. And in particular, I just wanted to know, what would you expect to happen in an animal or a human system treated with benzodiazepines, for example? Would you expect those learning responses and adaptations that you see to change in any way? And if so, in what way? Thank you.
Yeah, great question. Thank you. You've got to give me a few more years. We're just starting a program of work where we are combining animal and human work, using rodent work, using the same paradigms, the same visual learning paradigms to understand the role of GABAergic inhibition in learning. And one of the big questions actually is similar to BOLD. When the work started on the BOLD signal, we had questions about what does it correspond to. Does it correspond to spikes? Does it correspond to more complex signals? Now, thanks to seminal work in this area, we understand a bit better about the complexity of the BOLD signal. A similar thing is kind of happening with the MRS space and looking at neurotransmitters like GABA and glutamate. What is it actually that we measure with GABA when we measure it with MRS? How does it relate to GABA that we measure in animal models? And so this is exactly this program of work that we are about to embark on. We know that the synaptic GABA is going to be really hard to detect with MRI because simply the concentration is very small, the pool is small. We think that potentially what we are recording here is extracellular GABA, but we also know that that very much relates to synaptic GABA. So understanding the complexity of the GABA signal and in general the neurotransmitter signals that we are measuring with MR is really, really important for being able to then disentangle more the mechanisms. So that's literally where we are. It's a really fascinating question, and really looking forward to starting this program of work. We're planning to use biosensors that allow us to titrate what GABA we put in, and we'll be doing imaging in both rodents and humans with the same paradigms. So hopefully in a few years' time, it'll take time, and hopefully no pandemic.
Okay, there was here a final question, yeah? Can you bring the mic? So then the people can hear it during the recording. Yeah.
Hi, so I got a question about replication. One time someone asked Nancy Canwisher how many fMRI effects can be replicated, and she said way less than half, famously. So are you any more optimistic than that?
We are going from the nice and friendly to more ugly question.
Thank you, Richard. Well, Nancy is my hero, so
probably
nothing more
right.
to say on the topic. But am I more optimistic? I think we have made a lot of progress. We're also more sensitive in general in science into the importance of replication. But I also strongly think that there are no recipes in science. Science is about discovery. So it's really important, to keep an open mind and an open eye into interesting results. But it's interesting to be able to replicate. I mean, it's really important. Now, when I grew up with Ian and Suchita, our worst nightmare was not to replicate. It was the thing that woke me up at night, that my thesis won't replicate, that I presented at a conference and somebody will send me an email a few months later and say, I've tried the illusion you presented, and I don't see it. Nobody can see it, doesn't replicate. So I think it's really, really important to be sensitive to this. Now, the work I've shown you today, if you want, doesn't kind of look at new brain networks, doesn't discover, if you want, doesn't look on how from we can extract clusters in the brain that are involved in social decision making, or to give you an example, social kind of concepts or emotions. Or this work is absolutely valid, and it's really important there to consider individual variability, look at larger scale numbers. The work I presented to you today is very much driven from animal models and is looking at mechanisms. And I think depending on what questions we want to ask, we need to have the right approach, and we definitely need to be able to see that we can replicate. And actually, all of the work I've shown you, the GABA work, it's like the 4th time we are replicating these results. And every time that we can replicate, I actually feel, much more confident. The laminar work, the same. We have many more studies where it's really important to be careful with our methods, I believe, and also be able to publish protocols, procedures that we can follow. That doesn't mean to, stay and not have any innovation in the way we do science, but also to be able to document. And so I think we are learning a lot about how we do research at the same time that we are actually, being kind of immersing ourselves to these new methods. And I think that's where really the hope comes, that our work will be valid and will really contribute to our understanding and breakthroughs in science.
Mm-hmm. Okay, well, thank you very much, Zoe. And we are, thanks again.